Existing treatments for cancer often cause severe side effects. Chemotherapy, for example, focuses on cells that rapidly grow and divide. However, healthy cells can also grow and divide rapidly. It would be fantastic if we could destroy just the cancer cells.
So the current chemotherapy uses small molecules, and many of those small molecules that are administered in the patient, they go through the body and they kill cancer cells, but they also kill healthy cells. So that's a big problem. So therefore you have a huge tox to the patient. This limits the dose you can administer, and this limits the therapeutic benefit to the patient.
Marc Robillard developed chemically modified antibodies that can recognize tumor cells. Possible effects of this research are of course being considered within nanomedicine.
A solution for that is to target these small molecules specifically to cancer cells. And the way we do this is using a protein antibody. It's actually nanomedicine. These are nano-sized proteins that recognize cancer receptors. So these are targets on the outside of the cancer cell, specific to the cancer cell, and this can be bound by these proteins. So if you bind the chemotherapy to an antibody which goes to the cancer cell, and this way the chemotherapy is delivered to the cancer and not to healthy cells. We have devised a new way of going about it, where we have an antibody-drug conjugate. It has a linker holding both together, and after this ADC has bound the cancer cell to a receptor that's not fast enough to go inside the cell, we then cleave that linker ourselves by administering a probe in the second step. And this probe is injected, it finds the ADC on the outside of the cancer cell, it reacts with the linker between the antibody and the drug, and as a result the drug falls off and is then free to go inside the cell and kill the cell.
With this approach, you can target the chemotherapy at just the cancer cells, and this leads to a drastic reduction in the side effects experienced by the patient.